Good morning. Today I would like to talk about this paper called A Reliable Parameter to Standardize the Scoring of Stem Cell Spheres. A sphere formation assay has been successfully used for the isolation and enrichment of cancer stem cells in lung cancer, colorectal, and breast cancer. That's why in this paper, they decided to culture these cell lines. So here you have the cell lines that they use, a lung, a colon, and breast cancer cell lines. And they found significant heterogeneity in the size of tumor spheres from a variety of human cancer cell lines. We will see in this figure, in 8 days of culture, how the spheres derived from these cell lines, how they look. So some of them are very small here in the solid arrow, and these could be prone of subjectiveness to be classified as a sphere. And for example, the spheres that are derived from this cell line, which is a non-small cancer, a non-small cell lung carcinoma, these ones are round and they have sharp edges. So these ones would be easier to count. Whereas these colon cancer cells, the tumor spheres derived from these cell line, they have the tendency to fuse. That's why these tumor spheres are more difficult to be counted. And here they show the number of spheres versus the diameter of the spheres. And here you see the distribution. And all these variations in the morphological characteristic of spheres, along with the individual subjectivity to rule in or rule out a given small sphere, make it challenging for the precise sphere counting. That's why they propose this new parameter, the standardized sphere score, which is the summatory of the total volume of all the spheres. And here we see in this formula, divided by the total number of cells initially plated. Here you see in the formula, N is the total number of spheres and D is the diameter of the spheres. And what they show here is this staining in order to be able to see how single tumor cells are packed in the tumor sphere. So they introduced this new parameter to see how packed a sphere can be. And here we see how they, with the area, they draw a circle, and now they measure the radius and they are able to calculate the volume. And this new parameter that I told you is the sphere packing factor. And with that value, they saw that the standardized sphere score objectively reflects a stem cell potential. And this linearity that you see in these with these different cell lines, it shows that this linearity of the standardized sphere score is not cell line dependent. So for all the cell lines, we have this correlation index. It's very good. It shows 0.95 or 0.94 for the breast cancer cells, 0.94 for embryonic mouse lines. So this parameter, it has a good agreement with the idea. And also they show the application that this new parameter has with when comparing it when using different drugs. So they use, here are the drugs that they used, and according to the concentration, when the concentration of this drug increases, they see that this percentage of the standardized sphere score is a better parameter, whereas the percentage of the number of spheres doesn't show what is happening with the spheres. So here you see with the different drugs, the behavior and this percentage of standardized sphere score objectively reflects stem cell potential. So here they show in the table pros and cons of this parameter. So the sensitivity, it's more sensitive than other parameters in detecting changes in sphere forming ability. The accuracy, because it minimizes accounting error from identifying and measuring smaller spheres. And the adaptability, and it doesn't need to harvest the spheres, and the assessment at different time points and subsequent functional experiments or intact spheres. The cons are the morphology, because it requires a typical spherical structure, and it's time-consuming because to be able to count the 24-well plate that contains between 50 to 100 tumor spheres typically requires 30 to 60 minutes. And it is not applicable when significant fusion of spheres is present. Okay, thank you.